This video is for educational and informational purposes only. The content is based on personal experiences and is not medical advice. Always consult a qualified healthcare professional for personalized medical guidance.
So clearly we have an obesity epidemic, let's be honest. If people knew that obesity is replacing smoking as a major risk factor, but they don't know that. So there's a massive lack of knowledge, I think, on the part of the scientists who think cancer is a genetic disease, and they're constantly hunting for gene mutations that might be responsible for your dysregulated cell growth. There's a pharmaceutical industry making billions and billions of dollars coming out with immunotherapies or some other kind of therapy dependent in large part on the somatic mutation theory, which has now been undermined, discredited massively. And then you have the hospital industry, which generates tremendous revenue for radiation and chemo and this other stuff. And then you have the federal government that says cancer is a genetic disease, awarding research grants to investigators at top medical schools and science centers looking for mutations in genes that might be responsible for your dysregulated cell growth. So you have mis and disinformation at every level in the cancer industry. There's two issues. There's one, somatic mutations, which is called the somatic mutation theory. This is where mutations of lifestyle or living cause nuclear gene mutations. Then there's germline mutations. And you hear about the BRCA one with Angelina Jolie. She's made that kind of famous, having her breasts and ovaries removed or something like this. And then you have the Li-Fraumeni, the Lynch syndrome. These are germline mutations. And we have a paper just ready to submit with my colleague, Bob Kaplan, where we went through every major syndrome, inherited germline syndrome, and every one of those inherited mutations disrupts oxidative phosphorylation in the mitochondria. So, and none of them are 100% penetrant, meaning that a person could have the mutation, like BRCA1, it's like 60% penetrant, meaning 40% of the women that would have that mutation never develop cancer. They only develop cancer if the mutation disrupts oxidative phosphorylation, leading to substrate level phosphorylation and dysregulated cell growth. So there's secondary risk factors. The primary risk factor for all cancer, all major cancers, is insufficient oxidative phosphorylation coupled to a compensatory substrate level phosphorylation, leading to dysregulated cell growth. Not that complicated. So, but for a lot of people, it's overwhelmingly complicated. So you just have to know what the inherited risk factor is no different than if when we're exposed to a chemical carcinogen or oncogenic virus, hepatitis C, papillomaviruses. I showed how those viruses are linked to dysregulated cell growth through an effect on oxidative phosphorylation. So the virus or the product of the virus goes to the mitochondria, disrupts sufficient oxidative phosphorylation, leading to chronic upregulation of substrate level phosphorylation and dysregulated cell growth, driven by fermentation, glucose, and glutamine. Every major cancer is doing the same damn thing. So it's one disease. It's not 100 different diseases. The common pathophysiological mechanism is insufficient oxidative phosphorylation coupled to increased substrate level phosphorylation, facilitated by oncogenes, HIF1 alpha, MYC, open the floodgates to get glucose and glutamine into the cell. So those oncogenes are just facilitators of driving the substrate level phosphorylation. We have that paper that's going to come out in our big paper. So all of this is there. We publish it open access. Everybody is welcome to read it. If they choose to ignore it. So you can't do anything about that. Again, the origin can be one person could get breast cancer from a different reason from another person, but the bottom line is all the cells are fermenting. And regardless of what particular mechanism was responsible for the chronic disruption of oxidative phosphorylation, you're dealing with a population of cells that are fermenting. So now you have to deal with that, which is then goes into the management section. So we can try to avoid cancer by avoiding the provocative agents that would lead, but once you lead to disruption of OXPHOS. But once you have cancer, now you have to deal with the fuels that are driving it. Well, when you look at carcinogens, they call these molecules carcinogens, and because they cause cancer. And the question is, oh yeah, I see a lot of DNA mutations when I look at a tumor that was caused by a carcinogen. But the origin of how that dysregulated cell growth came in the first place If you look at, there was a beautiful study, I have it in my book, where they looked at these carcinogen exposed cells, and they're bioluminescent, they're fluorescent. The inside the mitochondria are like glittering, which is from the reactive oxygen species, and that's where the mutations largely come from. When oxidative phosphorylation is not efficient, ROS, reactive oxygen species, are the product of dysregulated oxidative phosphorylation, and these ROS are carcinogenic and mutagenic. They further damage the lipids and the proteins of the mitochondria, and they cause nuclear genetic instability and other kinds of mutations, as you see as downstream effects. And the cancer cell seems to be buffered from the ROS because a major reducer of reactive oxygen species is the glutathione system. And the glutathione system is part of the phosphate, the pentose phosphate pathway, producing a large amount of NADPH that drives this what we call antioxidant system. And the carbons from glucose and the amino acid glutamate are part of the glutathione system. So it's unbelievable. So the two fuels that are driving the dysregulated growth also protect the tumor cell from its own damage, from the damage of the ROS, but the nucleus of the cell is being damaged. So you're collecting DNA mutations as a downstream effect of oxidative phosphorylation, and people are all focusing on the downstream effects rather than the original, the fuel that's driving the beast. So once we know that, we know how to put out the fire. We know how to slowly degrade or interrupt the two fuels driving the dysregulated growth and keeping the rest of the body healthy. So when normal cells are burning ketone bodies, we produce much fewer ROS. And the bioenergetic mechanism of that was worked out with my late friend Richard Veach from the NIH, where ketone bodies do not produce FADH2. They produce only NADH, delivering electrons to complex 1 and keeping the coenzyme Q coupled in a fully oxidized state. Because that's where the ROS are coming from the half reduced form. And when you start throwing more and more ROS out, you have a tendency to degrade the efficiency of your energetic system. So burning ketone bodies is a way to protect the integrity of your mitochondria, maintaining a highly efficient energy state, keeping you from not only cancer, but numerous other chronic diseases. And also life. You live longer when you keep the ROS restricted. So it's aging, anti-aging, anti-cancer, anti-chronic disease, but nobody wants to do it. Right? This is as long as you know that. See, when you're completely unknowledgeable, then you would never know what I'm telling you. But now you actually know and have a mechanism that can explain it. Now it's your choice to say whether I want to engage or not engage in these activities. I haven't found one yet. So, and our papers show all major. So when I publish these cancers data on the damage to mitochondria, lipid drop accumulation, these are all biomarkers of dysregulated cell growth. These are all biomarkers of inefficient oxidative phosphorylation. 98% of all cancers fall into this. Is there a cancer out there that can grow out of control with normal oxidative phosphorylation and not dependent on substrate level phosphorylation? I've offered $100 to my students for the last 5 years, a crisp $100 bill. I'll give it to you. You find a tumor that can grow in a dysregulated way without a dependency on substrate level phosphorylation. I'll give you the $100, no problem.
Throw a Twinkie in, and they might be able to do it.
No, they ain't going to do it with a Twinkie. Because the Twinkie's loaded with sugar, you got to do it with a cancer cell that's respiring without fermentation and growing out of it. Now, interesting thing is liver regeneration can grow as fast. Regenerating liver cells can grow just as fast as tumor cells. So you have a hepatoma, which is a liver cancer. Okay, that will permeate your whole liver and kill you. Well, if you take a lobe off your liver, the liver naturally regenerates very rapidly. And they are using glucose and glutamine in very low quantities, but they're respiring those two fuels. They're not fermenting those two fuels. So it's very interesting to see that. And the liver knows when to stop. Liver regeneration knows. It just doesn't keep developing more and more new lobes of liver for yourself. They know when to stop. But the hepatoma does not know when to stop and ends up killing you. So, Kaplan, he's just an associate with us. He's doing he's working very hard. on this, but Christos Xenopoulos in Semmelweis University, the world leader on mitochondrial substrate level phosphorylation. When you don't know certain things, you reach out to those individuals that are the experts in those areas. You then introduce them to the problem. They start looking at it with their level of expertise, and then you bring yourself, the groups together to do this. We have a lot of physicians that are joining who are reading about what we're doing because they're into the issue of patient care. So they want to use ketogenic metabolic therapy to look at the outcome of their patients. But these are in small clinics and isolated conditions. They're not at the top medical schools, the top cancer research centers. They're not. These guys are all locked into the somatic mutation theory, lockstep with whatever the NIH says. So whatever the NIH says, whatever the standard of care says, they're in lockstep with that. And they're not willing or capable or even desirable to change their way of doing things. All of our funding comes from philanthropy and private foundations. Folks make the donations to the university, Boston College, saying, I want to support Seyfried's research. So we get donations directly because I cannot accept a nickel from anybody. It has to come through legitimate channels. The university has a channel here. And then we have private foundations, Travis Christofferson's Foundation for Cancer Metabolic Therapies. And then private foundations are reaching out slowly to us, because why should you dump in thousands and thousands, potentially millions of dollars into these programs that haven't done anything? There's no accountability. Where the hell does all the money go? Susan Komen, breast cancer found people running and swimming and jumping and climbing, and all we do is get more dead people. There doesn't seem. And then when you look at what they're doing, they're hunting for genes. And you say, now I know why you get all these dead people. The only people getting healthy are the ones running and jumping and whatever the foundation is, but the poor people are getting brutalized. So 1,700 people a day, and you can raise money for the lung cancer foundation, the breast cancer, the brain cancer, and then you do standard of care and get brutalized. So and they feel good raising money for a failed operation. Well, that's what our hope is. But we were slapped down at Cedars-Sinai. They wouldn't do it because Jethro Yu is working with us, and he said they'll do metabolic ketone only after you do standard of care. Well, standard of care is only put you on the slope to death. So now you're going to try to take and not only keep a guy alive, but try to correct all the damage that radiation and poisoning did to the body. This makes no sense. Why don't you start with metabolic therapy, and then if that doesn't do a real good job, you might want to consider a little temozolomide or something like that, rather than poisoning and irradiating the guy up front. Why don't you consider that as a secondary option after the tumor shrunk. I don't know what happened. Yeah, now you want to irradiate this poor guy when the tumor is shrinking and the surgeon can say, I think I can get it all out. I mean, everything is back-ass forward. And it takes knowledge. People have to become familiar with what I'm saying. And we're publishing this. It's not like I'm telling you this. Go and read the papers over and over again. Open access papers. We got them out there. People can read. Yeah, well, my book is Cancer is a Metabolic Disease. I wrote that book in 2012. Cancer as a metabolic disease. I speculated on some things. I had an idea about mitochondrial substrate level phosphorylation as being the second arm of the fermentation pathway. But then I got Christos Xenopoulos to work with me to prove it. And that paper came out in ASN Neuro for brain glioblastoma. But now we're starting to see it for all these different cancers. So it's just a matter of time. I don't know how long it's going to take, but we're not going anywhere. As long as philanthropy and private foundations support us, we're going to pound down this research. They choke on it. So you have no choice but to accept it. Well, I publish in peer-reviewed good journals. Cell and Science today have become overly involved with minutia. They love to have supplement after supplement after supplement. Oh, you didn't do this. We got 1,700 people a day dying. You're wasting your time on this kind of bullshit. Let me show. And then we got the Greek study. Oh, I can't believe that. We had this tremendous success. There must be something wrong with that study. But, my friend Greg Glassman, the former owner of CrossFit, he runs the Broken Science Initiative. We found that half the stuff in Science and Nature can't be reproduced. This is one of the great problems. Nobody can reproduce that crap. Nobody has enough time to look at. First thing you say is the study in Science and Nature in cancer based on the somatic mutation theory or the mitochondrial metabolic theory. Let's look at it that way right off the bat. If it's in the somatic mutation theory, get rid of it. It's not going to have any relevance to the clinic anyway. So then you start looking at the overall outcome in preclinical systems. How long did you keep your subjects alive? Were they syngeneic orthotopic models, meaning the tumor genetics and the host genetics are the same, not in these genetically created monstrosities that we're doing. So you have to look at the systems under which you're working. Well, as xenograft, you're taking a human cell and you're growing the human cell in a mouse that has a broken immune system, that has type 2 and type 1 diabetes. You got 50 million years of evolutionary difference between the biology of the human cell and the host mouse. And then you get some revelation. Whoa, look what happened. Then you put it in the clinic and it doesn't work. Those should be the last models you look at. The models you should look at are all natural ones, where the tumors arose in that strain of mouse, and it's a natural arising. See if you can manage those kinds of tumors, not some of these genetically engineered things. Yes, they're great technological improvements. For what, I don't know, but it's certainly not dropping the death rate. We got 1,700. If you knew what you were doing and you had a model that was really powerful, we wouldn't have 1,700 people a day dying. So that tells you something's wrong with whatever system, whatever program, whatever you're doing ain't working. And we know that metabolic therapy can drop that death rate very, very significantly. And we have the understanding of the mechanisms responsible for that. But it's just too radical right now. Too radical. So we got to wait for the field to catch up with what you know. So, but every one of those things that has a terribly different genetic or completely different, very different genetic background, they're all fermenters. So that's the commonality of all things. The common pathophysiological mechanism is what, as I said, the replacement of oxphos with substrate level phosphorylation, regardless of what the genetic profile tells us. Yeah, Mike Levin and I had discussions. As a matter of fact, I cited his work in my 2015 paper on metastasis, because metastatic cancer arises from fusion hybridization of our immune cell macrophage with a cancer stem cell. And that has the capacity to spread around your body, and you don't need new mutations for that. I told Michael that. He knows that. So we know what metastatic, and those metastatic cancer cells are glutamine driven. So they need the glutamine. So we know how to manage all the metastatic cancers. I mean, publish that. And listen, here's another thing. I published that called The Origin of Metastatic Cancer. That's my most highly cited paper, 1,230 citations that detail the origin of metastatic cancer as part of the immune system, right? Wow, you think that somebody would catch on to it? In the beginning of the paper, I said cancer metastasis is largely responsible for cancer deaths. That's all they read. So they cite the paper because I made that super stupid little set. Yet when you go to the ACR, American Association of Cancer Research meetings, row after row of people with the EMT, MET, EMT, I said, don't these people have any functional brain cells? This doesn't make any sense evolutionarily. If it's glutamine and glucose dependent, you can kill metastatic cancer. So we came to this revelation because I had a model of brain cancer from Edinburgh. It was called the VM. They formed natural tumors in the brain. And then I said, it took me my students and I over 10 years to figure out what the metastatic cell was. And it turned out to be a macrophage. And then we did a deep dive on I wrote a paper in that review, Origin of Metastatic Cancer, where my student, Leanne Heisentreit, we went through all metastatic cancers and saw that they manufacture markers of macrophages. Indicating that there was some sort of fusion hybrid condition. And Akil in 1905, a German pathologist, said he thinks metastasis comes from some sort of fusion hybrid. The late John Pawlik at Yale was jumping up and down, screaming and yelling about cancer being some sort of fusion hybrid condition. Melissa Wong from Oregon Health Science Center has been saying this. A number of people have been saying this. That there's the cancer has characteristics of immune cells, right? So and Candace Pert, a very powerful, recognized scientist, said the same thing, and she was vilified. And the attack, she published it in Science, saying that lung cancer is a macrophage kind of a disease. And there were people that attacked her, and boy, I'll tell you, she laid into those guys like there was no tomorrow. I give her a lot of credit. She just tore the ass off some of these idiots that were challenging her work. She was the leader in the field, and you didn't look at the data. So it was great to see that happening. In a cancer situation, you have a lot of inflammation. You have a lot of necrosis. You have a lot of mess there. And our immune system is designed to heal wounds. This is what we do. So if I cut myself and lead a big contusion, I've got hypoxia, I've got bacteria there, and then my monocytes come pouring out of the bloodstream and differentiate into macrophages to facilitate wound healing with fibroblasts and also killing bacteria that might be in there, because it's going to be a race for life or death. If the bacteria win, you die from sepsis, you die from bacterial infection. If your immune system wins, you kill the bacteria, you heal the wound, you got a scar, and you survive. Cancer is like an unhealed wound. Cancer is growing, creating the same microenvironment as you would have in a wound. The problem is the cells that are creating the disturbed microenvironment are fermenters, but they can't metastasize. They might be like some stem cell thing. So they grow, but they're throwing out acidified lactic acid and succinic acid, signaling the immune system to come in. The immune system comes in and throws out cytokines and growth factors, thinking they're facilitating wound healing, but it's actually making the situation worse. It's doing something evolutionarily programmed in a wrong context. So, and it gets worse and worse. Now, macrophages have a unique capability. They can fuse with each other to facilitate wound healing, or they can fuse with other cells in the microenvironment. So if I have a bunch of stem cells with already corrupted oxidative phosphorylation and mitochondria, and are fermenting, and the macrophage comes in and cytoplasmically fuses with the cytoplasm of the stem cell, all of a sudden you get a mixing and a dilution of the mitochondria in the macrophage. All of a sudden now you have a cell that can ferment, but now it's dysregulated because the organelle that's regulating that cell has now become part of the problem. So you have the most powerful cell in your body running rogue, but it has the genetic capability of suppressing normal immune cells, surviving in the circulation, called intravasation, where they leave the site, enter the bloodstream, go through the bloodstream, extravasate, and get into tissues spreading around the body, all because they are programmed to do this. The problem, the organelle that controls them doing it in the right way is now corrupted. And as long as they have fermentable fuels, they're rogue macrophages, and that's the origin of metastatic cancer. I spent 10 years doing this. They're part of our immune system. And you can kill them because we know what they eat. And that's why I tell you, first thing you do is, they're big. That's why I call them macrophage, big eater. Okay, so they eat stuff. So, and that's another thing too. When they're starving, they will eat each other. That's why you really have to be, and they'll eat the microenvironment stuff, and they'll eat microbeans. We tested all this stuff, so I know what they are, and I know how to kill them. The problem is I tell people that, and they look at me like, "Oh, I never heard of that before." Well, for Christ's sake, you're not doing the research like we are. You're not reading the papers like we are. Yeah, I mean, obviously, if I didn't care about it, I wouldn't have published any of that. I just keep it to myself. So I would know if I got metastatic cancer, I know exactly what to do, and I feel bad for everybody else who doesn't know what I know. And the thing you'd want is the person that would know this. So the team is the patient's knowledge and the physician's knowledge together, working together as a team to work with that individual, this didn't work so well for you. Let's just try that. But we're still putting pressure in this direction. So in other words, you have two knowledgeable individuals, and the outcome will always be significantly better than if either of them don't know anything, or one knows and the other one doesn't. So you've got to have a team effort in managing cancer, but it also will manage all these different chronic diseases as well. And when you consider the enormous fund of how much money our country and other countries are spending on diabetes, cardiovascular disease, Alzheimer's disease, cancer, where all of these can be managed basically with the knowledge of keeping your GKI low and bringing your body back into some sort of a Neolithic period, and recognizing those products of our great technology that can be greatly enjoyed under certain conditions. So it's going to be a lot of knowledge, because we're going to evolve. Who we are today, if we live, may not be who we are in a million years from now. We're going to know a lot of things that are called mysteries and unknown will become known if we can survive as a species. But knowing facts and replicating facts are the basis for the development of theories. So once you have a significant observations that are repeated over and over again, you develop a theory to explain that phenomenon. And as you begin to develop theories to explain the phenomenon, we move forward in what we can do. And this was the same thing with the heliocentric geocentric theory. It was the Copernican revolution that said it was the sun, not the earth. So you could explain the movement of the planets. You could explain things. You could better understand the universe based on these kinds of things. And this is the same thing. Once you understand the biology of cancer and you put the pieces together, you can move forward with disease management and understanding ourselves better. So this is the future. It's just that right now you have a lot of obstacles that need to be knocked down in order to move the field forward. But it's going. It's coming. Yeah. Well, this is the advance of science. This is the way. This is the new revelations that you see. These paradigm changes. This is the way it works for sure. So anyway, I'll let you guys go. I have a meeting that I have to get to, but get yourself a keto mojo. Start measuring your GKI and finding out what zone you guys are in. So when you go to a cocktail party, it now becomes a point of discussion. Oh, how's your GKI today? Well, whatever party you go to, whatever group you go to. But yeah, it's going to be a transformation, which is exciting. Whenever you have a new generalization, it makes people think about things they previously didn't think about. And let people know that if they want to support our research, they can support it at Boston College, because we depend on those folks, the philanthropists, the people who say, I don't really care if I make a buck on this. I just want to be part of the movement. And it's going to be exciting. So, or Travis Christofferson's foundation, these kinds of things, because that's how we keep going. So now after I have to go to this meeting, what I do is I have these same conversations with my group next door. And we talk about these things for hours and hours and hours, always looking for a fatal flaw in our concepts and trying to identify any blind side that we might get from some piece of evidence that might be contradictory to what we think. And then we evaluate it and we do experiments to test it. Okay, guys, have a nice day now.